What is the mechanism of HPV proteins causing cancer? Imagine a tiny virus that can hijack your body's own cells and turn them into cancer cells. That's what human papillomavirus, or HPV, can do. It's a common virus, but certain types are linked to cancers like cervical cancer. The key players in this process are two proteins the virus produces called E6 and E7. These proteins interfere with your cells, normal control systems, leading to uncontrolled growth and, eventually, cancer. E7 is like a saboteur that targets a protein called retinoblastoma, or PRB. Under normal circumstances, PRB acts as a brake on cell division, making sure cells only divide when they should. When E7 binds to PRB, it marks it for destruction. Without PRB to keep things in check, cells start dividing uncontrollably. This rapid growth is a hallmark of cancer. E6, on the other hand, targets a different tumor suppressor called P53. Think of P53 as the cell's quality control manager. It detects DNA damage and either halts cell division to allow for repair or triggers cell death if the damage is too severe. E6 binds to P53 and promotes its degradation through a process called ubiquitination. When P53 levels drop, cells with damaged DNA can survive and continue dividing, increasing the risk of mutations that lead to cancer. These viral proteins don't just act alone. They manipulate the cell's protein degradation system, called the ubiquitin proteome pathway, to destroy these critical tumor suppressors. By doing this, HPV effectively removes the cell's natural defenses against cancer. Beyond that, HPV oncoproteins can cause changes in how genes are turned on or off without changing the DNA sequence itself. They influence epigenetic modifications like DNA methylation and histone changes, which can silence genes that normally prevent tumor growth. This makes the cellular environment more conducive to cancer development. E6 and E7 also interact with genes involved in cell growth, such as C, MYC, and activate telomerase, an enzyme that maintains the ends of chromosomes called telomeres. Normally, telomeres shorten as cells divide, eventually stopping cell growth. But by activating telomerase, HPV-infected cells can keep dividing indefinitely, a key feature of cancer cells. Another important aspect is that HPV DNA can integrate into the host cell's genome. This integration can cause chromosomal instability, leading to mutations and structural changes in the DNA. Such instability accelerates the progression from normal cells to cancer. Finally, some studies suggest that HPV proteins can alter how cells use energy and even promote movement and invasion into surrounding tissues. This can help tumors grow larger and spread to other parts of the body. In summary, HPV causes cancer mainly through the actions of E6 and E7 proteins. They disable the cell's natural tumor suppressors, manipulate protein degradation pathways, induce epigenetic changes, activate growth-promoting genes, and cause genetic instability. These combined effects push the cell toward becoming cancerous. Understanding these mechanisms underscores the importance of HPV vaccination and regular screening to prevent these viral effects before they lead to cancer. If you have concerns or questions about HPV or related cancers, always consult healthcare professionals for personalized advice.